Hello, everyone. I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is A Panel Discussion, Genotyping by Sequencing for Novel SNP Discovery with Less Common Genomes, and our sponsor is LGC Biosearch Technologies. Our panelists today are Dr. Tiago Hori, Director of Aquaculture Innovation at Atlantic Aqua Farms, Nikolai Petra, Lead Scientist of Variety Development and Bioinformatics at Hopsteiner, Dr. Melissa Allen, Genomics Coordinator at the Center for Aquaculture Technologies, and Joris Parmentier, Global Strategic Marketing Manager at LGC. Mr. Parmentier will be leading a panel discussion with our experts, after which I'll have an opportunity to ask all of them questions submitted by you, our audience. You can type in a question at any time during the webinar. You can do this through our control panel, through our Q&A panel, which appears on the right side of the webinar screen. And if you look to the tray at the bottom of your window, you'll see a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Mr. Parmentier. Please go ahead.
Thank you, Christy. Appreciate the introduction, and I'll add my warm welcome to everyone today. I'm really looking forward to today's conversation. We've got a couple of great panelists on today. So without further ado, I'd love to start the conversation off for today. And I wanted to start with a conversation about the challenges of working with less common and complex genomes. And I'll actually start with Nikolai. And I'll start with hops, and although obviously important and certainly widely appreciated, definitely by me, it's probably not a genome as common as some of the typical row crops. I believe it's also a fairly complex genome with various ploidy states and in different variants, and also it has retrotransposons to deal with. I was hoping you can elaborate a bit on the challenges that has yielded for you during your time working with the species.
So yeah, hops is several papers have been published recently about the complexity of the hops genome. It's about 2.3 gig, but it is, I guess, similar to corn in its repetitive regions. It's about, say 90% repetitive, which makes it very difficult. And then in the GBS itself can make it complex because, where you think a tag might map to the genome, it could be mapping in several different areas. So one of the things that we've had to tackle using the hop genome is we've been able to normalize to different regions of the genome and leverage depth. And the genome itself, actually, the reference that's available right now is still in its infancy. Internally, there's a better genome at Hopsteiner, and then also the USDA is using a genome that was in collaboration with some other researchers. So it's getting better, and we're able to deconvolute some of these problems we have. But when working with a model crop, you have a lot of advantages over obviously working with a less common genome. But these are addressable, and you can overcome them using GBS with the right mindset, I guess is one way to put it.
And what do you mean with the right mindset? What sort of challenges do you have to overcome with that mindset? Yeah, can you hear me, Nikolai?
So, yeah, can you hear me?
Yeah.
Am I there? Okay, so with plants, you have chloroplasts, you have several, unmethylated repetitive regions. And, if you use, let's say, APK1, which is methylation sensitive, you can stay away from methylated regions, but there's still a lot of, let's say, like I said, chloroplastic unmethylated rDNA. And like I said, that can actually make it so, let's say, like 20 to 50% of your data can be, I wouldn't say useless, but less helpful. And so if you're able to do some additional sequencing or, I wouldn't say targeting, but I'd say enriching for regions of the genome that are actually useful, you can overcome, this 50% of your data being useless, and enrich for the parts that might be in linkage with, traits that you're looking for, if that's what you're using GBS for. Yeah, I don't know. It's a lot on the bioinformatics side, but also the ability to use the NCPI database and look for commonly known choroplastic regions and filter that out. You can get more useful data. I guess I'd maybe need to know, the depth of knowledge about GBS of the audience, but it's a enzyme targeted sequencing, and it started out, it's about 10 years ago, it was using APK1 because it was methylation sensitive, and typically is now using 2 different base pair cutters. And when you use these along with a little bit of sequencing, I mean, obviously you have to have you don't have to have your genome, sorry. Yeah, it takes a little bit more work, I guess, with the less common and more complex genomes. Yeah. That's for sure. And you need to know a little bit more, especially about your species. It's species specific to, if you're working in plants, animals, bacteria, it all is.
Yeah. No, absolutely.
Bioinformaticist, I know about hops. I can speak to hops specifically, but.
Absolutely. I think there's a bit of delay. Apologies for that. But no, I just wanted to confirm, Nikolai, that actually you could note also around the definition of the words, definition of the abbreviation. GBS is used widely as genotyping by sequencing, both targeted by primers as well as randomized by enzyme restriction. I guess today largely we're talking about SNP discovery applications, so that would refer to an enzyme restriction based GBS method. Thanks for that insight, Nikolai, very helpful. Switching, I guess, to fish or at least to aquaculture, I was hoping, Tiago, if you could help us understand some of the challenges that you've encountered. I know you've worked in aquaculture for a long time. Can you help us understand some of the challenges that you've ran into in the more complex genomes that you've worked with?
Yeah, I mean, I think that it's an interesting time for GBS because, back when we started, the biggest challenge is that you didn't have genomes. And that was the thing. And then you had to rely on unique or which is part of the Tassel pipeline, or stacks to call without a reference, just by piling up the reads and getting a reference feed. I think that has changed, and I think that is something that sometimes people don't realize. For GBS, depending on what you're doing, you don't need a fully contiguous genome. And with SQL2 around and the nanopore mini ion, it's not that hard to generate enough sequencing that you can that you can anchor your GBS data. So that's something that people should think more now, because our tendency is to think, oh, I have a obscure species, I'm never gonna get a genome, and that is not the case anymore. So that's one thing, is the absence of the genome. It's easier to solve now than it used to be. But when it comes to the complexity, you just have to know whether you're talking about a repetitive genome or a highly heterozygous genome or a highly polyploid genome. You will raise questions. So, for example, in salmon, the problem is because the genome is pseudotetraploid, and so you have incorrect mappings all the time, and you have to be able to use things like depth and quality. So one of the things that we've done a lot for species that when they have complicated genomes is that in several pipelines you can validate your SNPs by Mendelian inheritance. And I understand that there's various species that is hard to get some across, but a lot of species is not that hard. And so if you have the opportunity to get across, especially if you're in aquaculture or in agriculture, that shouldn't be a problem. Having a cross and being able at looking at Mendelian compatibility has been a way that really helped separate good from bad SNPs and false mappings. But I also think that you have to accept that you're gonna have to live with some data loss.
Yeah, understood. The data loss you're referring to, is that, can you quantify that a bit? So what is some data loss, and at what stage is it no longer acceptable?
Yeah, I think again, it depends, right? So if you're looking at doing things like genomic selection and GWAS, then you want coverage. You don't want to lose much more than maybe 20% of the discovered SNPs because you don't trust their quality, because you're just going to lose coverage. If you're doing diversity, you still want coverage, but you don't need fine-tuned coverage. You need more sparse places in the genome. So it depends on what you're targeting. That's why stacks, for example, is a favorite between the molecular ecology folks, because you can call without a hat reference and you're going to get enough to do your genome-wide diversity and FSCs and things like that. But if you're doing GWAS and things like that, and especially genomic selection, then you need coverage. And then you're going to have to go what Nikolai was saying, which is to try to maximize the usefulness of your data by using different enzymes and things like that, and understanding your genome.
Thank you. Yeah, appreciate that that extra background. Melissa, I guess some of the challenges that Tiago is flagging, things like heterozygosity, ploidy, repetitiveness of the genome, are those things that you've encountered as well?
Yeah, we encounter many of the same problems that Tiago was speaking about. So things like ploidy differences. Sturgeon, for example, can be tetraploid, they can be octaploid, they can be diploid. As Tiago was talking about, things like mollusks have extremely high levels of genomic polymorphism. We often work with Pacific white-sided shrimp, and they're known to have tons of repetitive junk DNA all throughout their genome. So when we were first working with them, we didn't have a reference genome available, so the bulk of the SNPs, when you would try to design them and further validate them, would fail. That species in particular now has a genome, but still, because of that complexity in their genome, you still get a higher failure rate for most of the SNPs. More recently, I've been working with coral species, actually, and a whole new problem has emerged that they have symbiotes in with them. So you have a mixture of DNA of symbiote and coral. So teasing those two apart has been especially challenging. And I'd say one of the biggest problems, you get a lot of just brand new species and just not knowing what the potential problems could be. Some are so understudied, you don't know if you're going to get problems with ploidy, heterozygosity, all of those things.
Thanks. That note around symbiote DNA and mixing, really interesting. How did you go about trying to, I guess, separate the two?
Well, none of the corals have genomes available or anything close to being helpful for that. So what I did was I used the symbiote genomes. So some of those were available, and one of them I was able to access prematurely before publication. So I actually used the symbiote genomes to try and filter out the GBS tags that were associated with them versus the coral. And we've done a first round of validation successfully for about 5 species so far.
That's great. Yeah, reverse engineering, I guess.
Yeah, can I just interject a second here? So there are 2, at least, quite widely used tools that are used for genome assembly, not so much for GBS, that you can use to decontaminate your data. One is Kraken, and the other one's BlockTools. But they're not dependent on the genome, but they are dependent on how many homologous proteins from your contamination are in the NR database on SwissProt. So you still have to have information, but you can rely more on closely related species because it uses BLAST instead of mapping. And so you can use these tools to blast and try to take the contaminants out. So that's something that I've done before, and it works, but sometimes it will overestimate contamination, and you have to be mindful of what you're taking out. But you can filter the reads using any of these tools before you go into your mapping step.
Thanks.
This is the normalization that I was speaking of.
That's in line with the
as well.
Go on, Nikolai.
Oh, I was just going to mention.
Thanks. Sorry,
we talked about the Mendelian segregation, and we found that. There's a 2019 paper that came out about hops, and they have quite regularly irregular Mendelian segregation. So we've had to actually rake out quite a bit of data. So not only mapping and blasting, like was mentioned, but also you have to find markers that behave correctly. And then one of the main problems also is heterozygosity undercalling, which there are some softwares and papers written about, but it's something you have to be cautious of. So with a smaller genome, depth can be, or not a smaller genome, sorry, a less common genome, depth can be very useful if you're just starting out, as opposed to getting as many, different accessions or taxa. It is beneficial to go a little bit deeper. And that's where the 2-enzyme thing comes in, just so you can be a little bit more confident in not having too much heterozygosity undercalling.
Got it. Yeah, and thanks, Nikolai, because I know you've been in several bioinformatics roles, and your earlier publications, your publications from 2013 on SNP detection in hops, for example, they were before the first assembly of the hops genome. I believe that was in 2015 somewhere. So I was wondering if you, and this is in line with the previous comments from Lisa and Tiago as well, I was wondering if you can describe some of the challenges that you ran into in your earlier work, particularly as it relates to data interpretation and bioinformatics for situations where you don't have a reference genome.
Right. So actually, Diego brought up the point of using, there's unique and stacks. And, this is pretty bioinformatics heavy conversation at this point, but, you have to know what you're looking for. As you mentioned, we actually used unique early on because of its, it helps with undercalling. It also helps with, not just blindly stacking genes, but it can use the entire tag as opposed to just the 64 base sequence. You can also bring in paired end reads to help with specificity of your SNP calling. And I know stacks is a favorite of evolutionary biologists and other things. I tend to stay away from stacks because it, from my understanding, literally just stacks things up, which can be useful, but in a repetitive genome or one with massive translocations, it can be misleading. So you got to do a little bit of homework on what do you want to achieve with your data, especially when you are lacking a genome.
Yeah, thanks, Nikolai. I appreciate that. And Tiago, you mentioned earlier that you've also worked with species without a well-referenced or annotated reference genome. I wanted to switch gears a little bit and ask around structural variation. Is that something you can get a good understanding of without a reference genome?
It depends, right? It depends on what you're talking about, structural variation, If you're talking about small deletions and small insertions, certainly you can. And one of the things that you can try to use in lieu of having a genome, if you're interested in structural variation, a lot of structural variation that has significance occurs in coding regions. So you can use a transcriptome rather than a genome, which is rather hot. It's easier to produce and assemble. So you can use that, and you can work with insertions and deletions that are small that will fit within a 150 or a 100 base pair read. You can use the traditional MPILOP protocol of SAM2s, which is not designed for GBS per se, but will work well to call indels. But the other side of it is that if you look at muscles, for example, we are seeing that the structural variation in muscles is sometimes the size of a gene, right? And you really only start to really appreciate that kind of variation when you're using some technologies like PacBio and Nanopore. And so I get no money from either Nanopore or PacBio, but I think that it's really now a time where people can really reach out to use GBS by pairing these two technologies, and then getting the better data possible. And if you're interested in structural variation, you really want to have, you don't need a genome, but get a bunch of scaffolds. 10,000 scaffolds doesn't matter. For GBS, it doesn't matter, right? If you get 10,000 scaffolds that are, 1,000 base pairs to 100,000 base pairs, that's perfect, right? If the SNPs are all that you're wanting, you don't need a fully contiguous genome. And if you look at, for example, even people will ask, well, but how do I determine coverage? So the Oyster folks in the UK did a chip for the Pacific Oyster without using GBS and without a contiguous genome, and they just used scaffold coverage and made their chip. And then when the Oyster's genome was scaffolded, long and behold, it was perfect. So again, understanding, I think this time where we are with the technology, understanding that you don't need a chromosomal level assembly. You don't even need a good assembly to start is really important moving forward, because it will prevent a lot of headaches that we had 10 years ago.
Thanks, Tiago. And I know, Melissa, you've obviously worked with a lot of less common species your time in aquaculture as well. What sort of challenges with regards specifically to data analysis and interpretation have you run into when it comes to these more complex genomes?
This is repetitive again, but finding the right pipeline to process it. And one of my favorites is the same as what's been mentioned. So tassel and stacks, they have unique and U stacks, respectively, to call SNPs in species with no reference genome. My personal favorite is unique, but I've also used U stacks as well. It can produce some bigger contigs. So depending on the platform you're trying to design to make a SNP panel, for example, which is one of the main things we do with GBS data, is make SNP panels. So you can use shorter tags, for example, if you're going for CASP assays by LGC, they'll accept, shorter flanking sequence. Sometimes you need larger, which is when I would move to U stacks. One of the biggest problems when you don't have a reference genome is you'll always get less SNPs compared to if you did have a reference genome. So for example, if you get 50,000 SNPs discovered when using with reference pipeline, you could get something like 30,000 if you use the exact same data set without a pipeline. I've actually tested this in one of the species of fish that I had that did have a reference genome. It was yellowtail jack. And I roughly got about the same numbers as what I saw in the literature. So yeah, you do have a drop in the number of SNPs you get. An additional source of SNP loss is I usually apply more stringent filters on the resulting SNPs than I normally would if you had a genome. So you end up with even fewer SNPs. So a lot of times this isn't a problem. When we're designing medium to low density panels, you still have plenty enough SNPs, but this could become an issue if you're trying to do GWAS or genomic selection. As Tiago was saying, for that you want higher coverage. But that's not to say you can't do it at all. We've been successful doing several GWAS studies for everything from growth, disease resistance. We've identified a few sex markers even through this method. The other biggest, I think, issue would be you have no idea where the SNPs are located and the proximity of them to each other. So like when you're designing a SNP panel, generally you want to spread them out across the genome. But if you don't have a genome, you kind of just have a bunch of SNPs and you don't really know where they are. Same goes if you're using them in a GWAS study. Often you'll build these fancy Manhattan plots, and you can kind of identify where a QTL is because you'll get this, peak of significant SNPs all in the same location on a chromosome. Whereas you don't necessarily have that when you don't have a genome because you have no locations. You just have a bunch of SNPs showing up as significant, and you don't know if they're close to each other or not. One of the things sometimes is we can use LD filtering when we're building our SNP panels to try and overcome this somewhat and try and get SNPs that are hopefully spaced out.
So help me understand that. How do you, and this will show my ignorance in genomic selection and other breeding approaches, but how do you use an LD panel to solve the issue that you don't know where the various SNPs are located in the genome because you don't have a genome? How does that work?
Well, once you get your SNPs discovered, if you do have a decently large population, when you're going through Plink, there are certain LD thinning features within Plink. Plink is a really common universal platform for processing and filtering SNP data. It even does some association analysis and things like that. So that's one method you can try to apply. But it still is going to be a downfall always of not having a genome. It's not exactly knowing where your SNPs are.
Got it. Hey, and before we move on from the topic of bioinformatics and data interpretation, Thiago and Nikolai, anything, for example, in what Melissa just referred to that you've ran into as well? Did you come up with the same sorts of solutions that Melissa did? Did you try any different approaches?
I mean, you have to try everything,
just
right?
Point out when we were talking
example,
about structural variation.
Thiago, if you go first.
You have to try everything, and I think that that's the point where understanding, the process and the technology is key. So we're moving towards a place in genomics now. There's more and more, and you have these plug and play interfaces and things that you can do with easy using things like Galaxy and other interfaces, but that can't preclude understanding the technology. Why do you use a certain restriction enzymes? Why does the dual restriction is, better? Why are you restricting at all? And then understanding anomalies in the data. Why does an area get over coverage? And even without a genome, you can see stacks that are way too deep that obviously look weird, right? And so, but you have to understand what are you doing, what is behind. You don't have to understand fully the statistics maybe, but you have to understand what the assumptions are and the things that are. And so I feel like sometimes we're moving away from that, and we can't. If you want to take the full power of this technology, especially in non-model species, you have to understand what the technology does and how it works so that you can identify the things that don't look proper and then make a decision on how you're going to approach that.
Yeah, thanks, Thiago. And before I hand over to Nikolai, just to follow up on that, Thiago, is that something you've, how did you, when we began working with this technology, obviously with our lab in Berlin, we've done a lot of work on this technology. Some of our scientists have as well. I know they're actually on the line today. And how did you, from your perspective, how did you go about generating that knowledge? I'm sure that's been a bumpy road to ensure that you really understood the methodology. How did you go about that? Did you work with other folks, institutes, collaborators a lot? How did that go?
You read a lot, you read the source papers and then you email the authors. You try to have a basic knowledge of statistics to understand what the assumptions are and genome biology. And I understand that not everybody's going to be able to do that. But what I would say is that if you're not going to go down that road and if you're not going to get the knowledge, then find someone to collaborate with that has that knowledge that can guide you and can help you. I know that not everybody has the time to learn all of this because in the end, I joke this, but Melissa knows this. And when we were working together, the way you learn is by breaking things, right? So I rewrote half of Tassel at one point, to see if it worked better, and it didn't. But you break it, and then you understand how it works.
Yeah, thanks. Nikolai, any comments on that topic?
I guess where this conversation started was the genome irregularities and structural irregularities, and I just wanted to point out that we actually used GBS to observe the non-Mendelian inheritance in hops, and found that there's permanent heterozygosity translocations, which then led us down the path where we started actually looking at the genome more carefully in its structural variation to try to uncover whether this was just a artifact of the sequencing or in fact it was true. And we published 2 papers in the past year actually about the structural variants. You can't say that I used GBS to explore this, but it did uncover it, using a few different methods, but mainly just, seeing the minor allele frequencies not behaving in a large swath of them behaving like we thought they would when sequencing a mapping family. And so just using GBS as I hate to say a Pandora's box, but you can analyze the data and really find some interesting things to continue to explore as well. That was just my 2 cents on the structural variation.
Thanks. Just another thing that I'll just add a little thing to that I would just add something to that with that is that it also depends on what you're trying to do. Because if you want to understand what the insertion of the deletion is, it's hard to have the full breadth of that with short reads. But if you're only trying to understand if it's there or not, then, for example, we are in mussels and in mollusks in general, there's a lot of presence absence variation. And we are actually looking at GBS data now to see if we can call hemizygous regions with confidence and use them to detect presence absence variants. So you can, right? But the problem is that it won't behave like your traditional SNP. You're not going to have a homozygous, heterozygous. You're going to have a homozygous, heterozygous, and you're going to have a hemizygous. Have not the presence of that region or that SNP. And so we are using it and trying to use it to see if we can confidentially then kind of detect the presence or absence of a certain structural variation with just short reads. It's to be determined if it's efficient, but you certainly can try.
Absolutely. Yeah. Thanks, all 3, for the comments on the topics around the bioinformatics and data interpretation. So trying to switch gears a little bit, and what I wanted to talk about was when do you use this? So I'm trying to think from the perspective of the folks on the line that may be starting to work with the genome that is very complex, that is very unknown. So at which stages during that process, and I'll ask all 3 of you, but let's start with Melissa. At which stages during that process would you say, now that is a great time to think about a method like GBS because XYZ benefits? So what advice would you give to folks that are starting to work on genomes that are not very well known, that are very complex? What advice would you share, Melissa?
Well, I'd say GBS is a really good starting point when you're working with something like that. So far it's been pretty reliable, especially if what you want to do is set up a SNP panel, for example. Then you can use that SNP panel to start, looking into population diversity and structure. If you're working with wild populations, even with aquaculture populations, we do this. So one of the big advantages I'd say is price. It's cheaper to do initial SNP discovery than almost any other method. Often when you have less studied species, there just isn't the funding there that there is with the bigger species. So that makes a big difference. You can pull off GBS projects at a much lower cost with much less individuals and get yourself, a decent amount of SNPs discovered. Also, GBS is, once you've gotten over the initial, learning curve of learning the programs, it's quite fast to process the data. Within 2 days, you can go from raw GBS data to a full-out designed SNP panel.
Thanks, Melissa. And Nikolai, from your experiences and thinking about the people on the line that are starting to work with these types of genomes, what would your advice be? Not just about when to apply GBS, but I guess more generally, what's the big challenges that the folks on the line can expect, and what sort of advice would you have to overcome that? Is this still delay? Are you still with us, Nikolai? Actually, let's switch. I'll ask you the question, Tiago. So same question as for Nikolai and for Melissa, I guess. So for those people here today with us that are starting to work with these novel genomes, more complex genomes, what's your key pieces of advice as they embark on that journey and start to explore these genomes?
I think that the most important thing is to understand your genome. And, even if it's just like going to a C-value database and see, well, what's the size of my genome? Is it half a gig? Is it 3 gigs? Is it 4 gigs? Or is it 15 gigs? Understanding that, then understand the evolutionary history of your individual or your MO. You don't have a reference genome, but maybe there is a closed species that has a reference genome. Is it highly heterozygous? Is it highly repetitive? Is it large? Is it small? You will be able to make assumptions about what to expect once you start to learn a little bit more about the genomes. For example, with mussels, we knew from the oysters that oysters have an incredible amount of heterozygosity, and we predicted that even though they're fairly distant bivalves, that mussels would also have that same level, and long and behold, it was, and you see for most bivalves. So understanding the evolutionary history of your individual can let you understand what to expect and what are the bumps in the road that you're going to face before you get them. Because if you get there and you don't know that, then you're a little lost.
Yeah, can imagine. I think we have Nikolai back, and if you're back, Nikolai, so what tricks that you know now would you really have loved to know 10 years ago when you started working on hops? Which of the tricks that you are applying now would have been very helpful if you would have known them then?
That's a good question. One of the things we're doing now is actually combining GBS with metabolomic data. And, this seems like an obvious phenotyping. You can hear me, yeah?
Yes.
Okay. Seems like an obvious phenotyping pathway, but, we've had some very encouraging things where, we've built, models to compare observations, let's say with disease resistance to metabolomic DNA that we've collected, and then, actually been able to bring in GBS and target these genes. When we start out, you make your SNPs, you run, tassel or GBS pipeline, and, you kind of you get an idea of where, what regions of the genome might be playing parts, what SNPs might be, in linkage with, something causative. And you can do that with mapping families. We've done that with several disease resistances, even yield characteristics, chemical profiles in hops. But the power of it to actually combine with some other technologies, like I said, like some metabolomic studies for, what is the molecule being produced and where in the genome is producing that. I think that it is more powerful than sort of just your, looking for maybe regions that correlate with a trait, but we were able to actually start parsing out specific causes, whether it's a promoter region or, and then you can take those pieces of the genome and look further. But I just was impressed with how well it does when you stack more accurate data on top of it, how it actually just gets more powerful.
And Melissa and Tiago, is that something you've done, stacking, for example, metabolomic data or other phenotypic data on top of your GBS data to improve accuracy?
We do. I mean, I've done several GWAS experiments where you have some level of phenotypic data. I think that the trick there, when Nikolai is taking, is that when you bring it down to the metabolomics level of a trait or of a phenotype, you're talking directly with the product of the gene or the causal gene. So the causal gene is an enzyme, is going to cause a change in metabolic. So that is way more accurate than if you were to try to correlate that with growth. And I think so. If you are going down the genome selection and the GWAS kind of road, you have also to understand that complex traits are complex for a reason. And getting down to the level of trying to tease out the complexity of what drives the trait sometimes is the only way that you'll be able to explain the variation on that trait. But sometimes that's not what you want, right? Sometimes you want the opposite because you don't want to measure 30 things. You want to predict growth. So it depends on where you're going. It really depends on what your goals are.
Right, it cuts both ways.
Yeah, no, I would agree with what Tiago and Nikolai have said regarding that. I mean, often we take the data and sometimes we'll work with it further down the line. And since we've worked it previously, more resources have come out that can help us, better analyze it.
Thanks. Hey, Tiago, before we move to the questions from the audience, one remark jumped out at me. Complex traits are complex for a reason. Help me understand why you said that.
So it's when you look at growth, for example, some people look at growth as if it is a trait. But growth is not a trait, right? Growth is a combination of disease resistance, metabolism, stress resistance, and infinite other things that contribute together to what you observe as a trait, and you call it growth, right? So that's a very complex trait. And so when you're trying to understand that trait on its own, sometimes it's very hard. I'll give you an example, is survival in shrimp. People have been trying to do markers for survival in shrimp, and it's very hard because survival is very complex. And it depends. Sometimes you survive because you're disease resistant. Sometimes you survive because you're stress resistant. Sometimes you survive because you're both. Sometimes you survive because you are in the top of the food chain. It's very hard. So sometimes you have to bring down to these small pieces that characterize what we call a larger trait to try to understand what is going on. And I mean, I'm sure that anybody that worked with disease resistance can tell you, even disease resistance is not really a trait. Disease resistance is a combination of millions of traits, and that's why it's very hard unless you have these rare times when you have a causal mutation, like MX in rats for orthomyxoviruses. But that's rare. Most of the disease are complex diseases, and they're going to be the causal SNPs are going to be all across the genome because it's not a single trait. You can argue about what is a trait, but what I'm trying to say is that all these things that we call traits, we should always refer to them as complex traits, not a trait, because they're not one thing. They're not one biological phenomena. They're the combination of many biological phenomena plus the environment. And so when you're doing GBS or any genotyping for that matter, for that kind of approach, you have to understand that also what the biological question that you're looking at. And that's one of them is that, a lot of the traits that a producer is going to tell when they want to select for are complex traits.
Yeah. Thank you, all three. Very
polygenic
much for,
traits.
Yeah, absolutely, Nikolai. Go
on. Sorry, I lost my train of thought. Give me a second. Oh, maybe I'll come back up in the questions. Sorry.
Not at all, and feel free to bring it up if it comes back. Thanks, all three of you, very much for the interaction. I've really enjoyed it. I guess, Christy, any questions?
Yes, we have a lot of questions, and thank you, Joris, for leading that fascinating panel discussion. As a quick reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to take a moment after the webinar has ended to give us feedback by taking our exit survey. All right, so let's jump right into our questions. So our first question is for all of our panelists. So what experimental design do you prefer? Do you prefer using a pan-genome reference or a population-specific reference? Let's start with Tiago.
I think depending on how heterozygous your genome is, a pan-genome approach is going to be too complicated, right? And so the question that I think is important is not so much what your reference is, but it was how informative the SNPs are across populations. And so I think rather than thinking about the reference genome, it's important to think that you capture the diversity of the populations you're interested for, so you don't select a bunch of SNPs that are going to turn out to be non-informative in other populations. And you see that happen all the time. And then you have a panel of 1,000 SNPs, and all of a sudden 500 of these SNPs are useless because they're all homozygous.
Melissa?
Yeah, I would agree with Tiago. If you have a population-specific reference, you would go with that for this type of application.
Nikolai?
I also just wanted to mention that, back to the accuracy of the phenotyping, if the trait wasn't specified in this question, but if you're able to measure a trait and see it segregate in a family, then it's obviously a great candidate for GBS and finding the marker, some causative marker and linkage. And if you're able to phenotype that accurately, but in other cases, you do have to kind of go germplasm wide if you can see, a certain trait spanning, well, maybe not germplasm wide, but, spanning an association panel. So it kind of depends on the trait, I would say, in addition to what the other panelists' input was.
Great. Yours, do you want to chime in as well?
No, we want to hear from the scientists, not from the marketeers, so absolutely not.
Great. Some of these questions will be specific to, or I'll address them to some of the panelists, but if any of you also want to jump in, please feel free. I should have said that at the top. Nikolai, to what extent do you think you can use genomes of closely related species to help you with bioinformatics analysis? And for example, if hops is closely related to cannabis, can you use cannabis to help you with bioinformatics analysis of hops?
So I would say yes as a starting point, but hops and cannabis are the closest related species in that clade. But the problem is hops' genome is about, I think, 6 times larger than cannabis. Cannabis is also an annual, where hops is perennial. I would say in general, especially I'm sure with the other scientists here, they can use closely related species. I think we just spoke about that with mussels, but plant species can diverge even quite quickly in evolutionary time. And so it really depends on what your ability to work with them in. But the USDA, John Henning, actually published a paper recently citing there are some there's some genes that are still held in common between them. So you definitely can use the crossovers, but you have to have a little bit more knowledge about them. And that's why in hops and in cannabis, there is some, and we do use the cannabis genome to some extent, but knowing how different it actually is does limit our ability to just be like, hey, look, these are both the same, so.
Great. Again, a question for all of you. What's your experience of using whole genome sequencing of pools of samples for SNP discovery? Melissa, do you want to start?
Yeah, sure. Just did this recently, actually, with the corals. But we don't rely entirely on pools. What we do is we have a couple of pools, and then we also have individuals singly run as well to validate that. But yeah, it's a useful method, especially like if you're doing an experiment and you're low on funds. It's a way of obviously getting more information and getting more individual sequenced, even though they're in a pool. But yeah, I would recommend, adding some single sequenced individuals into that mix as well.
Tiago?
Yeah, if you can, it works, right? The problem, one of the problems is that you don't get any sort of idea of frequency, or you get some, but and also you're gonna end up overestimating the heterozygosity in general. But I mean, if all your concern is that whether that the loci is polymorphic in a population or not to be used down the road, then yeah, by far. So I guess we didn't make the definition clear. There's a very strong differentiation between using GBS for discovery and use GBS for down applications, right? If you're going to analyze the GBS data with GWAS and whatever it is as it is, you have to be much more strict about what you're talking about. If you're just using it for discovery and those SNPs are going to go on a targeted GBS panel or on a PCR panel, on a Cas panel, what have you, then it's a whole different ballgame, right? And then pools and all of the things work beautifully.
And Nikolai?
Yeah, I hate to keep harping on, you got to know what you want to do, but, you'll lose a lot of data, pooling, you don't get any specificity of any, taxa. You might be able to parse out that one piece of data and have some confidence in it, if there's in a segregating population. It can be useful. I haven't used it too much because I feel like the cost benefit isn't quite there. You can use pooling and find out, one or two things maybe, but if you can get, the whole population and then use the modeling to parse out what you're looking for, but then you still have a swath of data that you can learn a lot from, where I think pooling can narrow that, even though it's quite a bit cheaper.
Tiago, you mentioned that there are tools for filtering out contaminants. Can you specify a little bit what tools that you were talking about?
Yeah, I think there's two that are classical. One's called blob tools, and the other one's called Kraken. They're all blast-based. I mean, in the end of the day, you could do it blasting your data. What it's doing is it blasting your reads against the NR database of NCBI or what have you, SwissProt, getting the annotations, and then also looking at the coverage on those annotations to try to figure out whether to try to assign a read to a clade. And so blob-tools, for example, give a list that these reads are assigned to this clade, and then you look at the clade, and if it's something that is completely related to your individual, you can easily remove that. In the case of chorus, for example, you could probably remove everything that is assigned to algae with quite a bit of confidence. In mussels, for example, because there's a handful of nematodes that live in mussels, we get a lot of nematode read DAGs. I mean, not a lot, but we get a significant amount in it. And so we take those out before we assemble the genomes. But realistically, you could do it with BLAST. It's just that either of these tools, either Kraken or blob tools, are kind of wrapped around. And blob tools has a little bit more of statistical sophistication, let's say.
So we have a question, again, for all of you. Can you comment on the fact that genomic selection accuracies of certain studies have reached a threshold at a low number of SNPs? Does that mean that that high density SNPs aren't necessary? Nikolai, what do you think?
Tiago, did you want to speak to that first? Sounded like you were going to say something.
Yeah.
Oh, no, it doesn't matter. I was going to say that depends, right? Because it's true. And so if you approach your genomic selection by trying to use your SNPs to increase the accuracy of how you calculate your relationships in your matrix, in your relationship matrix, either you're doing just a SNP-based matrix or a even better pedigree and SNP-based matrix. That's true. You can get a fairly small number of SNPs, increase the accuracy of your relationship calculation, and increase your accuracy. But you notice that that is the case for long-bred species, right? So species that you have a lot of pedigree data and they are not as outbred. I think that you will find that if you try to do that with outbred populations, you're going to suffer in accuracy. So again, it depends. You have to know what you're working for. You have to know what you're working with. But for the people here, if you're talking about species that are basically wild or very outbred, I think you're going to find hard to get good relationship matrices with a small number of SNPs, especially if you don't have pedigree.
Thanks, Tiago. I'm sorry, we have a little bit of a lag, so
right, and you don't know what.
Nikolai, please go ahead.
And you don't know what your SNPs are, in linkage and when you're, in linkage with when you're doing these outcrossing or wild species. So, you always need to be cognizant of, if this looks good in this, but what else is coming with it? And you can see this in, HOP in particular with the permanent heterozygosity translocation events. You might be selecting for disease, but or disease resistance or a higher chemical composition of something, but you could also be selecting for susceptibility to a different disease. There's a lot to parse out in populations like that. I just wanted to make sure that that point was made.
And Melissa?
I would say, too, you should look at the size of your genome, because that can have an impact as well. We've worked with some species with genomes so small that using a smaller SNP panel for genomic selection is actually feasible. Another thing to consider is also imputation. Now they're starting to say, like in salmon, for example, as little as 1,500 SNPs can be used to impute back up to a panel of 50K or 40K, 30K, and that can be a useful tool as well.
Great. And there was a discussion at one point between the three of you about about SNP failure. And we have a question about what SNP failure could look like and what that means in terms of what the data could look like when you see a failed SNP. So could you talk a little bit about that, Melissa? Why don't you take that one first?
So not sure what the question, if it's, SNP failure after the fact when you put it on a panel and try and genotype it.
Sure. Yeah.
Yeah. And that's also can be very species dependent. I think I was talking about shrimp earlier, that oftentimes because there's so much repetitive DNA, when you go to actually design the primers and create a panel, you do get SNP failure. And they're known for having null alleles as well. So that's something to consider if you know some sort of information about their genome, and if those could be issues.
Tiago?
That's the one side of the panel that goes down. But if you're looking at your GBS data and you don't look at it carefully, one of the things that you see is that you're going to see bias in frequency and bias in Hardy-Weinberg equilibrium. And both of those things are going to affect any results that you go down the line. So, one of the things that you can do is a filtering in frequency and filtering in a certain, it's a game, because obviously you don't want to filter everything that is not in equilibrium, but things that are in very significant equilibrium are either too close together to matter, or most likely they are an artifact. So it's really important when you're using the GBS data as your end data to identify these SNPs that are probably not real, because if you go with them to your analysis, they're going to skew your analysis completely.
And Nikolai?
I agree with what they said. Yeah, I mentioned heterozygosity undercalling. There's a good paper in Grapes by I think Katie Heimo is a lead author on that. And it's a very hard thing to parse out of GBS data without more depth. But across populations, certain anomalies have been observed that you can clean up your GBS data. And what I mean by this is, GBS might call, a certain SNP A when it's actually AT just because it lacks the depth to get both. So that's heterozygosity undercalling. And I found it to be actually, kind of a big problem in the heterozygous populations and species I work with. So I don't know, just referring you to literature might not be the most helpful thing, but definitely something you need to take into consideration if we're talking about failed SNPs on that side of the equation.
All right. Thank you very much. I'm afraid we have run out of time for today, so we're going to wrap it up there. We'd like to thank Tiago Hori, Nikolai Petra, Melissa Allen, Joris Parmentier, and our sponsor, LGC Biosearch Technologies. If we didn't have time to get to your question, we will try to follow up with our experts. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.